Hi everyone, welcome back for the second part of neurophysiology. Some more study tips. Talk to yourself out loud as you explain a topic. Speak for at least 2 minutes about what you know. You can record this and check your answers. You can do it with the study group, but it's really helpful to do something like this out loud because it's a lot harder to lie or cheat to yourself and say, oh yeah, I know this material, right? You have to explain it and explain it like you're teaching it. It can be really helpful. And so in our last section of this neurophysiology, we talked about action potentials and single neuron function. And today we're going to talk about how neurons talk to each other. Okay, so let's imagine here's our neuron of interest, and there are connections from other neurons that come down their axons and attach to the dendrite. You'll note in this picture that there's something called a presynaptic axon, and this is the postsynaptic cell then, okay? What that means, why it's pre and post synapse, is that there's something called a synapse, a little space here where neurotransmitters are released, and that tells these neurons how to function. And so we'll talk about the synapses a lot today. What I want to point out from this picture is that integration, so the connections of all of these presynaptic axons that come and contract the dendrites here, integrate their signals at the cell body, particularly this axon hillock segment. And all of those signals from other neurons will help this neuron decide whether to fire or not fire. Okay, so we're going to be talking today about EPSPs that are excitatory and IPSPs that are inhibitory. And only if this neuron fires an action potential will it release neurotransmitter from its axons on the dendrites of later cells. So let's start with the synapse. The synapse is how two neurons communicate. The first neuron here is the presynaptic neuron, and it's called a terminal bouton. So this terminal bouton, or synaptic knob, is what reaches out and gets really close to that second neuron. And there are many terminal boutons per neuron. The second neuron is the postsynaptic neuron, and the most sensitive region for this is the axon initial segment. Okay, and that's because it has a really high concentration of potassium and sodium channels. So a lot of ability to control that cell's action potentials. So this is what a synapse looks like. Here's our presynaptic cell. The synapse itself is the space here. It's called a synaptic cleft. Okay, and then the postsynaptic cell. And what you'll see is that neurotransmitter is released from our presynaptic cell when the action potential comes down the axon. And eventually that neurotransmitter will bind to the postsynaptic cell. We'll talk about mechanisms of binding in a little bit. So first we want to look at how neurotransmitter is released into the synaptic cleft. So an action potential comes down this axon. It reaches the axon terminals. And at the axon terminals, the voltage-gated calcium channels are open. So now we have a new ion we're thinking about in terms of membrane potential. Voltage-gated calcium channels open, and when they do, calcium rushes into your cell. Now remember, calcium is positive. Incoming calcium is going to raise the membrane potential of this area, but more importantly, it's going to cause this docking mechanism to take place and neurotransmitter exocytosis. Okay, so calcium is going to come in. It's going to bind to a sensor protein in the cytoplasm, and that creates a complex, a calcium protein complex, that makes the vesicles full of neurotransmitter dock and bind to the cell membrane of this presynaptic cell. And that releases the neurotransmitter then into the synaptic cleft. Okay, so again, showing it to you in words, action potential reaches the knob, voltage-gated calcium channels open, calcium comes into the cell, and binds to the sensor protein in the cytoplasm called a synaptotagmin. The calcium synaptotagmin complex releases the snare proteins that hold the synaptic vesicles in place, and exocytosis can occur. So vesicles filled with neurotransmitters fuse with the plasma membrane of the axon, and those neurotransmitters then are released into the synaptic cleft. They can then bind to receptors on the postsynaptic neuron. And there are 3 ways in which they can bind, or 3 different kinds of things they can do to bind to postsynaptic cells. We're going to talk about ligand-operated channels, G-protein-operated channels, and second messenger system. So in ligand-operated channels, the neurotransmitter is the ligand. So here's an example of acetylcholine as a neurotransmitter binding to ion channels directly. So they're ligand-operated because the ligand, the neurotransmitter, the thing it's binding, is what's causing the channels to activate. So here we have a channel activating, and they can be either sodium channels or potassium channels, but they are then going to cause ion movement across the membrane. Okay. So an example of a ligand-operated channel operated by acetylcholine, we call this ACH for short. That acetylcholine is going to cross the cleft. Two molecules are going to bind directly to the ion channel. The ion channel opens, and then ion movement can occur. Okay. Acetylcholine can also bind to a G protein operated channel. Now the difference here is that it's not binding directly to the ion channel. It's binding to a receptor that is coupled to G proteins. So when this neurotransmitter, when the acetylcholine binds, this receptor is activated and recruits the G proteins. Those activate the ion channel. And in this case, we're allowing potassium to come out. Remember, potassium's leaving the cell, you're losing positive ion. So this is going to decrease the potential of the membrane of this cell. Okay. So same thing in the beginning, acetylcholine crosses the cleft. When it binds to a protein receptor, it's not binding to the channel, right? It's binding to the protein receptor, which then causes the G proteins to activate and dissociate. And the subunits of those are going to bind to and open the ion channel. Now our third type is the second messenger system. Here we have our neurotransmitter, in this case it's norepinephrine, binding to a receptor. Again, we have G proteins involved, so the G proteins are going to dissociate. But in this case, they are going to activate adenylate cyclase. Okay, and this is an enzyme that's membrane bound. When adenylate cyclase is activated, it converts ATP to cyclic AMP, and that activated cyclic AMP activates protein kinases, which in turn open ion channels. So it's a longer pathway, and those protein kinases are also going to phosphorylate other proteins and can even do things like change gene expression. So we have an increase in this signal and a broadening of the signal by having this longer pathway. Okay, so when norepinephrine crosses the cleft, it binds to the protein receptor. The G proteins are activated and dissociate. And subunit signals adenylate cyclase. Okay, so remember this ASE ending indicates it's an enzyme. In this case, it's a membrane-bound enzyme that converts ATP to cyclic AMP. Cyclic AMP activates our protein kinase, and again, protein kinase is an enzyme. So what's it going to do? It's going to phosphorylate ion channels, so it'll open, and it can do a bunch of other stuff too. Okay, here's this action of our enzyme adenylate cyclase to convert ATP to cyclic AMP. Basically, you have ATP, adenosine triphosphate, right? 1, 2, 3 phosphate molecules on there. And adenylate cyclase encourages the removal of 2 phosphates. So cyclic AMP is adenosine with a single phosphate on there. Okay. So the neurotransmitters will continue to bind to their receptors and signal unless they're removed in some way. Sometimes there are enzymes in the synaptic cleft itself that can degrade the neurotransmitter. Things like acetylcholinesterase. So this is an esterase, an enzyme that is going to break down acetylcholine. Or neurotransmitters can be recycled back into the terminal from where they came. Okay, so there's 2 ways then of halting this signal, stopping that synapse. Okay, so here's showing you how the acetylcholinesterase works, where the acetylcholinesterase receptor is here and then breaks up this into acetate and choline. Okay. Okay. So lots of different kinds of neurotransmitters. You don't need to know all of them, but I do want you to know the basic categories and then look up an example of each one. Okay. So some neurotransmitters are amines that are derived from tryptophan. Some are catecholamines, where they're derived from tyrosine. And there are other amino acids as well. So GABA is one of them. It's really common in the brain. It's a very common inhibitory neurotransmitter. And there are polypeptide neurotransmitters. These are short proteins. Some of these are also hormones, okay? So they can act as hormones and they can act as neurotransmitters in the brain. And then there are neurotransmitters that are gases. These are ones that are going to be dissolved in the blood. And I want you to, again, know these categories and then look up some examples of each type. Okay, so how do we excite a neuron? Well, if we're going to excite a neuron, we need to create an excitatory postsynaptic potential called an EPSP. We can do this by increasing the membrane potential of our postsynaptic cell, and we're going to do that by opening sodium channels. Remember, you open a sodium channel, sodium is going to rush into your cell, increasing membrane potential. This is going to make our cell more likely to fire. Okay, so we're getting membrane depolarization, and actually tend to need more than one EPSP to actually reach the threshold so that that postsynaptic neuron will have an action potential. Okay, so let's think about that threshold value then, again, at negative 55 millivolts. We have our cell here. That's the one we're measuring. We have the cell here resting at negative 70. And now we have one presynaptic neuron releasing neurotransmitter onto our cell that's going to cause sodium channels to open. And so this cell is going to have a little bit of an influx of sodium. So that happens again. Then when we get this one and this one both telling this cell to open sodium channels, we get a much larger influx of sodium, a bigger depolarization, enough to get us to threshold that will then cause this cell to have an action potential. Okay, so that's called spatial summation because you have more than one neuron creating an EPSP on this cell such that it will reach threshold and have an action potential. Okay, so that was spatial summation. EPSPs from different neurons add together to reach threshold. In temporal summation, this happens in time. So you can get several EPSPs from the same neuron, and that will cause threshold to be reached. Okay, so how do we inhibit neurons? If opening sodium channels causes an influx of sodium that increases membrane potential, an inhibition of neurons is going to be IPSPs, and we need to have a way to decrease membrane potential. So how do we do that? We do it with either chloride or potassium channels. Because if you open chloride channels, you're going to get an influx of chloride, so an influx of a negative ion is going to decrease membrane potential. Or you can have neurotransmitters open potassium channels. When you open potassium channels, this positive potassium ion will leave the cell, efflux, and both of those will hyperpolarize or bring down the potential of the membrane, make it more negative. When this happens, then threshold's not reached. So our postsynaptic cell will be less likely to fire. It will not have an action potential. It will be inhibited by either the influx of chloride or the efflux of potassium. So sometimes you'll get an IPSP from one neuron and EPSP from another neuron. Okay? In that case, they summate and it's going to result in nothing. They're going to add together to make zero, no change, and cancel out. So here we are. We have an IPSP from one neuron and an EPSP from one other neuron. And as long as the IPSPs and EPSPs all go up and down and stay below threshold, our postsynaptic cell is never going to fire. Okay, so there's a lot to unpack here. I think commonly videos are really helpful. So there's lots of animations here for you to work through and look at and get a feel for all these different topics. But for the neurophysiology chapter, we need to focus on action potentials, particularly the movement of those ions, sodium, potassium, and chloride. Synapses, so how neurons communicate through synapses. And I also want you to read a little bit about and watch a little bit about graded potentials, so how this might work to actually cause more nuance in the brain than just firing or not firing. And so please take advantage of these, come talk to me in office hours and review all of this so that we're clear on how neurons function.